Take the tallest mountain on Earth, all 849 meters of Mount Everest, drop it into the Pacific Ocean, watch it sink, watch it fall past the sunlit zone, past the twilight zone, past the midnight zone where the last trace of solar light disappears and absolute darkness begins. Watch the mountain continue to sink for another 2 kilometers before its summit finally touches the bottom of the Mariana Trench. The ocean floor swallows it completely. There is still more than a mile of water above the peak. The Mariana Trench descends to 10 to 135 meters. That number is so large it stops meaning anything until you translate it into physics. And at the surface, the ocean pushes on your body with the weight of the atmosphere above it, roughly 14.7 pounds per square inch.
Manageable.
Survivable. At Challenger Deep, the lowest confirmed point in the trench, the pressure reaches approximately 16,000 pounds per square inch. Yeah, that is more than 1,000 times the pressure at sea level. It is enough to crush a steel submarine if the hull geometry is wrong. It is enough to compress the volume of seawater itself into a measurably denser configuration. It is a physical environment so extreme that the laws governing how matter behaves begin to operate outside of anything the human body has evolved to process or understand. No human being has ever stood on the floor of the Mariana Trench unprotected. No human being ever will. The pressure would not drown you. It would not merely crush you. It would compress your body cavities, force gas out of solution in your blood, and restructure your tissues at a molecular level before you registered that anything had gone wrong. You would not die slowly. You would simply stop being a coherent physical system, and yet something lives there, not one thing, not a handful of hardy microbes clinging to the edges of survivability, an entire ecosystem, fish, crustaceans, invertebrates, organisms that have solved in exquisite and sometimes horrifying mechanical detail every engineering problem that the deepest ocean presents. They are not monsters. That word carries the wrong implication. The implication that something exceptional and anomalous produced them. What produced them is the same process that produced everything else alive on this planet. An evolutionary pressure applied without mercy across geological time in the most demanding physical environment the Earth has to offer. These are not monsters. They are optimal engineering solutions to impossible conditions. The difference matters, because understanding how they work tells us something that no amount of surface-level biology can reach. It tells us what life actually is, stripped of every comfort, every redundancy, every system that evolution maintains, only because the environment has not yet punished its existence. At 11,000 meters, there are no luxuries. Every structure serves a function. Every metabolic pathway earns its energy cost. Every adaptation exists because the alternative was extinction. This is the Mariana Trench, and this is what it built. The first problem the deep ocean presents is mechanical. And the solution evolution arrived at is, by the standards of surface biology, deeply counterintuitive. Bones are a structural investment. In the context of surface life, they make obvious sense. They provide rigid scaffolding for muscle attachment, protection for soft organs, leverage for movement through environments where the body must work against gravity. Bone is expensive to produce metabolically, but the returns justify the cost. Across hundreds of millions of years and in virtually every major vertebrate lineage, bone has been refined into one of the most successful structural materials in the history of multicellular life. Pressure destroys that logic. Under extreme hydrostatic pressure, rigid structures become failure points. A bone does not compress uniformly under 16,000 pounds per square inch. It resists. And structures that resist pressure gradients without distributing that force through their geometry, develop stress concentrations at their weakest points. Those weak points fail. In engineering terms, this is called fracture propagation. In biological terms, it is called death. The deep ocean did not eliminate bone. It made bone irrelevant through a more elegant solution it selected for organisms that have so little rigid structure that there is nothing for the pressure to act against unevenly. The pressure becomes ambient. It becomes the medium, not the threat. Pseudoliparis suaire, the Mariana snailfish, is the deepest dwelling vertebrate ever recorded. Specimens have been observed and captured at depths exceeding 8,000 meters. It is, by any surface biology standard, barely a fish. The body is translucent. The skin is thin and fragile to the point of tearing under normal handling. The bones, where they exist, are reduced and flexible rather than dense and rigid. The skull is incompletely ossified, meaning the brain case is more membranous than structural. The whole organism is built around a design principle that sounds paradoxical until you understand the physics in order to survive a pressure that would destroy conventional structure, build as little structure as possible. The gelatinous body solves the pressure problem through passive compliance. When the surrounding water exerts force equally in all directions, a compliant body transmits that force equally rather than resisting it. There is no pressure differential to exploit, no structural weak points to propagate failure through. The organism is, in the most literal sense, part of the fluid environment. The boundary between organism and ocean becomes, at the mechanical level, almost irrelevant. This is why deep-sea creatures brought to the surface do not survive. And this is where the paradox sharpens into something genuinely disturbing. The same structural compliance that makes a snailfish mechanically stable at 8,000 meters makes it catastrophically unstable at the surface. Removed from the pressure that holds its diffuse body in equilibrium, the organism degrades. Tissues that function perfectly at depth begin to lose integrity in conditions that surface biology considers normal. The animal is not killed by the surface environment in the way that a surface animal is killed by the deep. It simply ceases to be structurally coherent. It falls apart. The engineering trade-off is absolute. The solution to surviving the deep ocean is a body design that cannot survive anywhere else. There is no intermediate form that functions at both extremes. The pressure environment of the Mariana Trench does not merely select for adaptation. It selects for organisms that are permanently, irreversibly committed to the conditions that produce them. Evolution built a perfect machine for one environment, and that machine cannot exist in any other. Below approximately 1,000 meters, solar photons cease to exist in any biologically meaningful quantity. This is not dim light. It is not the diffuse glow of an overcast sky or the faint illumination of a moonless night. It is the complete absence of electromagnetic radiation in the visible spectrum. Nothing that relies on reflected sunlight to navigate, hunt, or communicate has any function at these depths. The visual systems that dominate surface ecology, the image-forming eyes of fish and mammals and birds refined across hundreds of millions of years, are in absolute terms useless. But the deep ocean did not eliminate vision, it transformed it. Macropina microstoma, commonly called the barley fish, lives in the mesopelagic and bathypelagic zones and has solved the light scarcity problem with a piece of optical engineering that has no direct equivalent in surface vertebrate biology. The fish has two large tubular eyes oriented upward within a transparent fluid-filled dome that constitutes the upper surface of its head. These eyes are not designed to form broad images of the surrounding environment. They are designed to do one specific thing with extraordinary efficiency: detect the faint silhouettes of prey animals passing above against the dim residual light that filters down from shallower water. The tubular geometry maximizes the light gathering capacity relative to the retinal area. It is a biological telephoto lens, trading wide field of view for sensitivity at low photon densities. The transparent dome that surrounds the eyes protects the delicate optical structures while allowing unobstructed lines of sight in multiple directions simultaneously. The entire system is built around the constraint of operating in an environment where photon scarcity is the dominant selective pressure on any visual system. But other organisms have gone further, much further. Malacosteus niger, the stoplight loosejaw, has solved a more sophisticated version of the light problem. Most deep-sea bioluminescence is produced in the blue-green range of the spectrum, around 470 to 490 nanometers. This is not an accident. Blue-green light penetrates seawater more efficiently than other wavelengths, and the visual systems of most deep-sea organisms are tuned to detect it. In the deep ocean, blue-green is effectively the only color that exists. Everything else is filtered out by the water column long before it reaches abyssal depths. Malacosteus niger produces red light. Specifically far-red bioluminescence in the range of 700 nanometers, well outside the detection range of most deep-sea predators and prey. This is a private communication channel built into the body of a fish. The stoplight loosejaw illuminates its prey with wavelengths that the prey cannot detect. The prey does not know it is being observed. It does not know it is being targeted. The loosejaw, which has evolved specialized photoreceptors capable of detecting its own red output, moves through a darkness that is absolute for everything else but functional for it alone. It has not merely adapted to the absence of light. It has created a sensory monopoly in an environment where no other organism can operate on the same frequency. And then there is the broader phenomenon that underlies all of this. Bioluminescence. In the deep ocean, it is not a curiosity. It is not decorative. It is the only light that exists, and it serves every function that sunlight serves on the surface: predator attraction, prey detection, mate signaling, species recognition, threat signaling, and counter-illumination camouflage. Estimates suggest that upward of 75% of deep-sea organisms produce some form of bioluminescence. In the Hadal zone, the transition from rare capability to near-universal trait is essentially complete. An environment without sunlight did not eliminate light. It replaced sunlight with a more efficient, more targeted, more controllable substitute that organisms could produce direct and modulate in real time. The deep ocean removed the input and forced biology to rebuild the system from the output side. What the deep ocean did to vision is not impoverishment, it is redesign. In the sunlit surface ocean, photosynthesis produces the base of every food chain. Solar energy drives the conversion of inorganic carbon into organic compounds. Those compounds are consumed, concentrated, and transferred up through trophic levels in a system that is energy rich at the base. Even where individual organisms are scarce, the fundamental input, solar radiation, is effectively unlimited. None of that applies in the deep ocean. Photosynthesis ends at roughly 200 meters. Below that depth, every calorie that sustains life in the water column has to arrive from above. The mechanism is called marine snow, and the name carries a romantic inaccuracy that the physical reality does not support. Marine snow is not delicate or picturesque. It is a continuous slow rain of biological detritus dead cells, fecal pellets, shed exoskeletons, aggregated microorganisms, decaying fragments of organisms from every depth in the water column above. It takes weeks to sink from the surface to abyssal depths. During that descent, it is consumed, processed, and metabolized repeatedly. By the time it reaches the Hadal zone, the fraction of the original surface productivity that remains is a small percentage of what began the journey. The deep ocean runs on leftovers. Specifically, it runs on the leftovers of the leftovers of the leftovers of a surface ecosystem that is itself not particularly generous with its waste. This energy poverty is the master constraint that shapes everything else about deep-sea ecology. When food is scarce and arrival is unpredictable, organisms cannot afford metabolic strategies built around consistent caloric input. The deep ocean does not select for fast growth, high reproductive rates, or energy-intensive activity. It selects for patience, efficiency, and the physiological capacity to wait. And then, occasionally, something extraordinary happens. A whale dies. A whale fall, the descent of a large cetacean carcass to the seafloor, is one of the most significant ecological events that can occur in the deep ocean. A single large blue whale represents approximately 40 to 60 tons of organic material. At depth, that carcass does not decompose quickly. It becomes a temporary ecosystem, a concentrated pulse of energy in an environment defined by energy scarcity. The colonization sequence has been documented in detail. Mobile scavengers arrive first, hagfish, and sleeper sharks and crustaceans, consuming the soft tissue over months. Then enrichment opportunists colonize the exposed skeleton, organisms that exploit the nutrients released from decaying lipids. Then, in a final stage that can last decades, chemosynthetic bacteria colonize the bones themselves, breaking down the lipid-rich structures and supporting a community of organisms functionally analogous to those found at hydrothermal vents. A single whale carcass can sustain an active deep-sea ecosystem for 50 to 100 years. It is a city built on a dead animal, lasting longer than many human institutions, sustained entirely by the chemical energy locked in its bones. The organisms that exploit these resources have adapted to the feast or famine structure of deep-sea energy availability in ways that produce some of the most anatomically extreme designs in the animal kingdom. Deep-sea gigantism is one of the more counterintuitive results. Giant isopods, crustaceans related to common pillbugs found in garden soil grow to lengths of 45 centimeters or more in the deep ocean. Deep-sea amphipods, which are shrimp-like organisms typically measured in millimeters in shallow water, reach lengths of 20 to 34 centimeters in the Hadal zone. The leading explanations for this size increase involve metabolic efficiency at low temperatures, reduced predation pressure, and the ability of larger body size to sustain longer periods of starvation between feeding opportunities. A larger body has more internal reserves. In an environment where meals are separated by weeks or months, more reserves mean longer survival. But the most extreme energy efficiency adaptation in the deep ocean belongs to a group of fish whose reproductive strategy sounds from the outside like a biological horror story. The deep-sea anglerfish, specifically the solution it evolved to the problem of finding a mate in an environment of near-total darkness and extremely low population densities. In the deep ocean, encountering another member of your own species is a statistically unlikely event. For a species that reproduces sexually, this creates a fundamental problem. How do you find a mate when the probability of any encounter is so low that waiting for another opportunity might mean waiting longer than your organism is designed to survive? The anglerfish did not evolve a courtship ritual, it evolved fusion. When a male anglerfish encounters a female, he bites into her body. His immune system, which would normally reject foreign tissue, is suppressed at the point of contact. His circulatory system fuses with hers. Over weeks, his body degrades. His eyes go first, then most of his organs. Eventually, what remains of the male is a small protrusion on the female's body, retained testes connected to her bloodstream, receiving her hormonal signals and releasing sperm when her body produces eggs. A single female can carry multiple males simultaneously. This is not parasitism in the conventional sense. It is a reproductive architecture designed for the worst-case scenario in a worst-case environment. The male does not need to find a second female. He does not need to survive independently. He does not need to do anything except exist as a functional reproductive organ attached to a female who handles all other survival requirements for both of them. In an environment where the probability of re-encountering a mate after separation is effectively zero, the strategy that maximizes reproductive success is the one that makes separation impossible. The deep ocean made the anglerfish's reproduction look like a nightmare. What it actually is a logical conclusion. In 2019, a research team conducting a survey of Challenger Deep, the lowest point in the Mariana Trench, recovered footage and samples from depths exceeding 10,000 meters. Among the material documented at the bottom of the deepest place on Earth was a plastic bag, not a fragment, a bag, identifiable, intact, drifting in the water column at the maximum depth of the world's ocean. The implication of this single observation requires a moment of precise consideration. The Mariana Trench is geographically remote. The nearest major landmass is hundreds of kilometers away. The trench is thousands of meters below the reach of conventional shipping or fishing activity. Getting a piece of human-produced material to Challenger Deep requires that it travel from wherever it was discarded into a river or directly into the ocean through the surface current systems that circulate across the Pacific Basin, and then sink through more than 10,000 meters of water column over a period of years. The plastic bag at the bottom of the Mariana Trench did not get there by accident in any local sense. It got there because the systems that transport human waste through the global ocean are more efficient than the systems that contain it. The bag is the visible part of a larger problem that is considerably less visible. Microplastics, polymer fragments smaller than 5 millimeters produced by the degradation of larger plastic items have been documented in deep sea sediments at depths exceeding 6,000 meters throughout the world's oceans. In the Mariana Trench specifically, studies published in the last decade have found microplastic contamination in the sediment at Challenger Deep, and more significantly, inside the bodies of deep-sea crustaceans recovered from hadal depths, amphipods collected from the Mariana Trench, organisms that have never been closer to the surface than several thousand meters, contain synthetic fibers and plastic particles in their digestive systems. The contamination pathway runs through marine snow. The microplastics in the surface ocean aggregate with the same biological detritus that forms marine snow. They are incorporated into the same sinking particles. They descend through the same water column. They arrive at the same seafloor that receives everything the surface ocean discards. The deep ocean's energy import system, its only connection to the caloric resources it cannot produce itself, is also its contamination import system. There is no mechanism by which the deep ocean can filter one from the other. The contamination problem exists alongside an emerging industrial threat that operates at a different scale: deep sea mining. The seafloor of the world's oceans contains polymetallic nodules, rounded concretions of manganese, nickel, cobalt, copper, and other commercially valuable metals that form over millions of years at abyssal depths. The Clarion-Clipperton Zone, a region of the Pacific roughly half the size of the continental United States, contains nodule deposits estimated to contain more nickel, cobalt, and manganese than all land-based reserves combined. The demand for these metals, driven by battery technology for electric vehicles and renewable energy storage, has generated significant commercial interest in extracting them. The extraction process, as currently proposed, involves bottom trawling machinery that vacuums nodules from the seafloor and pumps the material to surface vessels. The sediment plumes generated by this process extend hundreds of kilometers from the extraction site. They smother organisms on the seafloor. They introduce fine particulates into the water column in quantities that have no precedent in deep sea ecology. The nodules themselves, which take 5 to 15 million years to form, would be non-renewable on any human timescale. The organisms that live on and among them, species that have not yet been cataloged or even discovered in many cases, would be eliminated from those areas permanently. The deep sea ecosystems at risk from mining are not understood. They are barely observed. The Hadal Zone has received less systematic scientific attention than the surface of Mars. The number of species documented from depths below 6,000 meters is a fraction of what almost certainly exists. Scientific expeditions to the deepest parts of the ocean remain technically difficult, extremely expensive, and logistically complex enough that the entire history of direct hadal observation spans only a few decades and a small number of research programs. We are proposing to industrially extract material from systems we have not characterized, using processes whose ecological effects we cannot model with confidence in environments where recovery timescales exceed any reasonable human planning horizon. The core problem with deep-sea mining is not that it is necessarily impossible to do responsibly. It is that we currently lack the scientific baseline to know what responsible means in this context. We cannot quantify what we would be destroying because we have not yet determined what is there. The plastic bag at Challenger Deep is not the most significant thing humans have done to the deep ocean. It is the most visible. The more significant things are invisible, embedded in sediment, incorporated into tissue, and still in the planning stages of extraction proposals that are advancing faster than the science that would allow us to evaluate their consequences. We are moving into these systems before we understand them. The history of every other ecosystem humans have approached that way is not encouraging. Consider the snailfish again, the translucent, gelatinous, apparently fragile organism that holds the depth record for vertebrate life. By any surface biology aesthetic, it is not impressive. It has no dramatic hunting adaptations, no bioluminescent lure, no fused reproductive appendages. It is pale and boneless and structurally minimal. If you described it to someone without showing them an image, they might reasonably question whether it qualifies as a fish in any meaningful sense. But consider what it is actually doing. It is surviving actively and successfully in an environment where the pressure is sufficient to crush titanium alloy vessels if their engineering tolerances are insufficient. It is finding food in an environment where the caloric density of the water is orders of magnitude lower than in any surface ecosystem. It is navigating, reproducing, and maintaining metabolic function in complete darkness at temperatures near the freezing point of seawater. And it is doing all of this with a body architecture so precisely calibrated to those conditions that it cannot exist anywhere else. The snailfish did not compromise, it optimized. That is what evolution under extreme constraint produces, not generalists that can tolerate a range of conditions. Specialists so perfectly fitted to a single set of conditions that they become expressions of those conditions in biological form. The stoplight loose jaw is not strange. It is a logical conclusion. Given an environment where all conventional visible light is absent, given prey animals with visual systems tuned to blue-green wavelengths, given the physical possibility of producing red bioluminescence through modified photophores, the trajectory that leads to an organism hunting in its own private frequency band is not random. It is the outcome you would predict if you ran the selection pressures long enough at sufficient population sizes across enough generations. The loose jaw is what you get when evolution solves a communications engineering problem. In an environment where the solution space is constrained but not closed, every organism in the deep ocean is a case study in constraint-driven engineering. The anglerfish's reproductive fusion is not grotesque. It is the minimum viable solution to the problem of sexual reproduction in a low-density population distributed across a three-dimensional dark environment with no reliable meeting points. The giant isopod's body size is not anomalous. It is the optimal configuration for a scavenger whose food supply is unpredictable and whose metabolic expenditure at low temperatures must be minimized. The barrelized transparent dome and upward pointing eyes are not exotic. They are the predictable outcome of optimizing a visual system for maximum photon capture in a specific directional range under extreme light scarcity. What the Mariana Trench reveals with a clarity that no other environment on Earth provides is what evolution actually is. Not a process that produces variety for its own sake. Not a mechanism that generates complexity through accumulated refinement. A process of ruthless, indifferent engineering under constraint, which produces organisms that fit their environments so precisely that they and their environments are, in a meaningful sense, the same thing. The snailfish is not a fish that lives in the deep ocean. The snailfish is what the deep ocean built when it applied the rules of natural selection to vertebrate body plans over geological time. The distinction sounds philosophical. It is actually physical. Understanding the deep ocean matters for reasons that extend well beyond the immediate practical concerns of mining regulation and contamination monitoring. The deep ocean is the largest habitat on Earth by volume. It covers more area than all of the planet's land surfaces combined. It has been continuously inhabited for longer than complex life has existed on land. The organisms it contains represent evolutionary lineages of extraordinary antiquity, adapted to conditions so stable over geological time that their design solutions are essentially fixed. They are experiments that ran to completion. We have explored less than 5% of it. Let that number sit for a moment. Every discovery described in this video, the snailfish at 8,000 meters, the stoplight, loose jaws, private light frequency, the whale fall ecosystems that last a century, the anglerfish's fused reproductive architecture, all of it comes from less than 5% of the accessible volume of the deep ocean. The remaining 95% has not been systematically observed by any scientific instrument. We do not know what lives there. We do not know how many species remain undiscovered. We do not know what engineering solutions evolution has produced in the Hadal zones of the Atlantic and Indian Oceans, in the deepest reaches of the Arctic Basin, in the trenches of the South Pacific that receive even fewer scientific expeditions than the Mariana. The Mariana Trench is not special because it is the deepest. It is special because it is the only part of the deep ocean we have studied in enough detail to begin to understand. It is the keyhole through which we have glimpsed a system that occupies the majority of the planet's biosphere. The creatures of the deep are not grotesque. They are not nightmarish. They are not alien, except in the superficial sense that their solutions to biological problems look unfamiliar to eyes calibrated by surface life. They are what life looks like when the constraints are absolute and the engineering has had millions of years to converge on optimal solutions. And they are also what we will never get back if we destroy the environment that produced them before we understand what it produced. The pressure at Challenger Deep is 16,000 pounds per square inch. The water temperature is just above freezing. There is no light, there is almost no food. By every metric that surface biology uses to define a habitable environment, Challenger Deep is incompatible with life. Life is there anyway. That fact alone tells you something fundamental about what life is. Not a fragile property of comfortable environments, not a phenomenon that requires warmth and light and caloric abundance, a set of solutions, adaptive, replicating, self-sustaining solutions to the problem of persisting in a physical universe that is, on average, deeply hostile to the existence of complex organized matter. The deep ocean did not break the rules of biology. It revealed that the rules are more permissive than anything that lives on the surface is equipped to recognize. We have mapped less than a quarter of the seafloor at the resolution needed to identify major geological features. We have sampled biological communities in the Hadal zone from a handful of trenches in the Pacific. We have conducted sustained observational studies at depth for accumulated periods that amount to days when summed across the entire history of human deep-sea exploration. Everything we know about the Mariana Trench, everything described here, represents the first sentence of a document whose length we cannot estimate because we have not yet counted the pages. The Mariana Trench breeds the creepiest creatures on the planet because it breeds the most precise. In an environment where every metabolic watt must be justified and every structural gram must earn its cost, evolution does not tolerate approximation. It produces, through the elimination of everything that does not work, organisms that are indistinguishable from the problems they solved. They are not alive in spite of the conditions. They are the conditions expressed in protein and membrane and chemical gradient. That is not creepy. That is engineering at a scale and precision we have not come close to matching. The question is not whether we can understand it. The question is whether we will give ourselves enough time to try.